Thank you for coming to our seminar. And we are going to talk about profiling of immune receptors, T cell and B cell receptors, and immunophenotyping of the T and B cells. So you probably already know that adaptive immune system is really very critical for understanding of wide range of the human disease, and most typical application for analysis adaptive immune system it's cancer, infection disease, autoimmune disease, food indifferent, allergy, organ transplantation, vaccine development. But applications can be more than shown on this slide. And definitely you can develop novel therapies, cell-based therapies like CAR-T, ACT, ADC vaccines for cancer. But also it's a lot of application for biomarker discovery, monitoring of the ongoing therapy or selection of the patient or best responders for the clinical trials. In order to give you introduction about what does mean TCR BCR, I would like to remind you that it's the most complex genetic system created by nature, and it's generated all this huge assortment of the T cell and B cell receptor generated in the course of genetic recombination in the immune cells. And complexity of this system is enormous. And as demonstrated here, at the end of the recombination, you have very similar structure of T cell receptors, B cell receptors, antibodies, which are organized very similar way, with all these circles which show CDR3 region, which is recognized foreign antigens, mainly recognized foreign antigens, CDR3, and so which presented by MHC peptide complex or directly by proteins. And what is interesting from genetic point of view, number of different T cell and B cell receptors and T and B cells which present in our circulation is several hundred million, and each cell has different T cell or B cell receptor. As a result, you can realize how complex is this system in comparison, for example, with number of human genes. Number of different T cell and B cell receptors at least 1,000-fold more than number of the human genes. It's create really significant problem in analysis, understanding of the population, how to profile it, and how to understand what is the antigen-specific T cells or B cells present in the human's blood, for example, or in any tumor samples. And if you think from biology point of view, this one is universal diagnostic system which created by nature, which allow you to detect any pathology or foreign microorganisms and really even eradicate these changes by really activation of T cells or B cells which try to kill all these not normal cells or foreign organisms. And we are going to talk today, I will give you technical introduction how to do immunophenotyping of the T and B cells and profiling of adaptive immune receptors. In order to facilitate these studies, we developed three technologies shown here. First, it's bulk profiling, adaptive immune receptor repertoire profiling, AACR assay for simplicity, which allow to profile starting from RNA or genomic DNA in a big number of the cells and generate very high quality quantitative data. Also, we developed immune marker assay for 500 top T and B cell markers, and single cell assay, which allowed to profiling 30 key cell type cell markers, and also identify full-length sequence of T cell and B cell receptors. And not just to give you in general idea, bulk profiling is definitely great if you need to profile dozens or hundreds of the samples. Single cell analysis gives you more detailed information about phenotype of the cells at the single cell level. But in terms of cost, it's significantly more expensive than running bulk analysis. Immune cell fraction analysis, where you do exactly the same protocol but try to work with the cell fractions, is somewhere in the middle. It's more cost effective, but also not as quantitative as bulk analysis. Let's start from bulk analysis of T cell and B cell receptors. We just developed these two assays based on the RNA or DNA. They are organized the same way. It's multiplex PCR using mixture set of the highly validated PCR primers. Actually, we don't use them as a PCR primers. We use them only for primary extension. And you can amplify, like shown on this slide, only CDR3 region or full-length region of RNA or DNA. We designed for both human and mouse, and what great, you can really combine all 7 chains in one reaction. So you can profile in one simple reaction all 7 chains for T-cell receptors and B-cell receptors. You don't need really to split them, and assay characterized by very high sensitivity and specificity. Importantly, using QFUMI facilitates significantly quantitative analysis of different clonotypes in bulk profiling data, and data analysis done using AACR program from my labs. This software is really one of the best software probably which is available right now for quantitative analysis in order to really generate reliable data for immune repertoires. So if you try to run this assay in bulk, for example, using 50 nanogram of total RNA from whole blood or PBMC, you generate number of the clonotypes shown here. It's approximately, you identify maybe 100,000 of different clonotypes for all chains. TRD and TRG, it's gamma delta, they are less abundant, so that's why you generate for them significantly less number of clonotypes. The problem is, results are very, if you try to repeat this experiment 3 times, for example, for 50 nanogram, and try to compare result, you will see very low overlap between 3 sets of the data, which is completely different from expression profiling. And what is the problem? Problem is that, for example, here it's like experiment done and replicates for 50 nanogram of total RNA, whole blood RNA, and you see from 200,000 identified clonotypes, 199,000 clonotypes are not reproducible at all because they amplified, if you see on the y-axis, it's number of the starting RNA molecules, because we can count exactly how many molecules starting population using universal molecular indexes. And you can see practically all clonotypes you identify amplified from single RNA molecule. Definitely it's unreproducible if you try to do it in triplicates. But if you only focus on the clonotypes which are present at more than 10 copies, you see these guys which show by arrows 20%. In this case, data will be reproducible. So this one is show you what is the problem in data analysis. If you need to focus on something which is present at least 10 molecules of RNA in the set of bulk data. In this case, you can, if you just focus only on the top clonotypes and hits top 100 clonotypes, you can see it's very reproducible. You start from 25 or 100 nanogram, you can generate the same data. Or even you can start from dried blood microsamples, which is 30 microliters of the blood, like shown here on the left side of this image. So in this case, you generate reproducible data. What great, you can generate the same data if you use just tumor samples without even purification of T and B cells infiltrating lymphocytes. Because technology is very sensitive, and up to 50-fold excess of other cell types still allow you specifically amplify only immune receptors from lymphocyte population of the tumor samples. And not to talk only about technology, I will give you one example, and it's a result of our collaboration with Professor Wilson Hudson from Emory University. And this scientist tried to identify T cells and B cells which are activated in metastatic tumor samples in brain lesions. So what they done, they take from multiple patient samples, you see, purified T and B cells from brain metastatic lesions by fax, purified DNA and RNA, and they give DNA to adaptive biosystem, and RNA they give to us. Both companies did expression immune repertoire profiling, and you can see here definitely RNA show sometimes 2-3 times more clonotypes identify. And the question is, okay, you identify all these clonotypes, what to do with these clonotypes? Okay, you identify 10,000 clonotypes per sample. What you're going to do with 10,000 T-cell receptors or B-cell receptors? So we speculate that if you compare RNA and DNA data, it allows you to identify clonotypes which can be activated in these T and B cells which present in the tumors. And because if the cells are activated, T-cell receptors and B-cell receptors can be activated in terms of transcriptional level. By normalization of RNA level to DNA level, you can identify activated clonotypes. And here's the comparison which show identification of these predicted activated clonotypes. And I would like to put your attention to this right part, where is the IGH, you see? Here it's R, it's data generated from RNA. D, it's generated from DNA. And this one is abundance level of the different clonotypes, overlapping clonotypes between DNA and RNA data. And you can see these guys interesting clonotypes. For example, 5% of the clonotypes, like shown here, you see, has 1,000-fold more number of copy of the RNA than DNA. So it means they have 1,000-fold activation. It's rather obvious to assume that these clonotypes correspond to plasma cells, which actively produce antibodies in these B cells, activated plasma cells. The same true, you can see for T cell receptors. It's left part of this slide. And you can see here we have about approximately 10-20% where you have not so dramatic 1,000-fold, but at least 50-fold activation of transcription for several clonotypes. It shows you power of this technology when you combine RNA and DNA analysis for the same samples. But analysis of clonotypes, it's only first step in the journey in order just to develop something interesting data. You need to characterize the cells which has specific interesting clonotypes. In order to do it, we developed a panel of the 500 immune marker. And this guy, we spent here significant efforts. We just screen all literature available, public databases, publication, and identify 3,000 candidate genes, which are really very critical for immunology, especially of T cell and B cell immunology. And we screen all these, design multiple primers, screen them against panel of 40 immune cell types, activated PBMC and whole blood cell, and develop final set, the most informative 500 genes like shown here. We believe that this type of assay can be used in combination with single cells or bulk profiling or profiling in immune cell fractions in order to get some information about phenotype of the cells. And logic of this type of the screens is following. For example, on the top you can see here it's 10x strategy. So you take tissue samples, dissociated or blood samples, encapsulate cells in the droplets, like here on the top, and generate from each cell expression profile together with the sequence of T cell receptor or B cell receptor which can be present in this cell. After that, what you're doing so-called T-SNP plot, where you cluster transcriptional profiles based on the similarity to each other. And you, for example, you have cluster of naive cells, memory cells, or exhausted cells for T cells. After that, you really generate data which show you TNB biomarkers and T cell receptor sequences. Interesting that you can do very similar strategy using more simple approach. You can immediately sort the cells based on the same biomarkers which characterize naive cells, memory cells, or exhausted cells in the test tube. And you can run this immune marker assay, what I described right now, and error assay using the same cells. It allows you to generate very similar set of the data. We also described today a single cell assay, so you can sort cells in 96-level plate and do very similar strategy. But number of the cells definitely will be limited, not like in 10x, 10,000 cells, but it can be a few 100,000 cells. And I would like to show you a short example, small example how it can be used. Okay, we sorted cells for different type of the CD4, CD8, naive, memory, effector cells, and run both assays what I described. It's air profiling in order to characterize clonotypes and immunophenotyping of these cell fractions. And the data looks like shown on the next slide. And you can see here, on the left side, you can see some markers for the each sorted cell fractions, what we are sorted, and set of the clonotypes which correspond each of the phenotype on the left side. What great in this technology, you can do assay directly in sorted cells. You don't even need to purify RNA. You can directly use cells in the assay. It's not necessary to purify RNA. And second, you can combine together phenotypic characteristic of the cells, specific cell fractions, with the set of clonotypes which present in these cells. A couple of words about single cell analysis. And we believe that this technology is rather simple alternative to 10x profiling, VDJ profiling. And what you are doing here, you just take the population of the cells and sort them in 96-well plate. For some application, you don't need really to do global analysis of all immune cells which present in your biological sample. For example, if you're interested, for example, to analyze activated cells, you can sort the cells using, for example, activated T-cell markers like CD133 or CD25. Usually the number of the cells which are activated in, for example, PBMC sample or even in tumor-infiltrated leukocytes, it's rather small, like shown here on the FACS image on the top. It's maybe only a couple hundred cells. In this case, you don't need really to run expensive assay. You just sort them in the plate, put in -80 refrigerator, and can do this analysis at any time convenient for you. Or even combine multiple plates together from different experiments and run together. This assay is very simple. You just run assay, after that combine all wells together and run as a one test tube. So it's very simple strategy. It's not something new. It's what people do already for the last 10 years. It can be also cell, if you're interested in some cell, for example, which recognize specific antigen, for example, epitope, like MART1, for example, yes, for immunotherapy. Okay, you can really label cells by this dextramer or tetramer, and also analyze what is the characteristics, phenotype of the cells, and identify T-cell receptor which recognize this peptide. It also can be used for definitely specific cell fractions like exhausted T-cells. So as a summary, what we discussed today and try to give you a journey from, we believe that first bulk profiling is the first really assay what makes sense to do when you try to identify intrinsic set of the clonotypes which important really to run for follow-up studies. And after that, you can definitely make more deep characterization of the specific T-cell or B-cell receptors by running single-cell analysis or analysis in immune cell fractions. But definitely for single-cell analysis in immune cell fractions, you need to have alive cells. You cannot use RNA or DNA extracted from biological sample. We believe that single-cell assay, what I discussed right now, it can be good alternative strategy, cost-effective, and very user-friendly in order to run this type of analysis. And I would like to summarize what we discussed today in my short presentation. First, we discussed development of adaptive immune receptor profiling assay, which is designed for bulk profiling from comprehensive profiling of all 7 chains in single multiplex PCR reactions, and can be run from RNA or DNA. We discussed some simple strategy for detection of antigen-activated clonotypes by comparison of the RNA and DNA, rRNA and rDNA data. We discussed strategy for development of the assay for immunophenotyping of the T and B cells using set of 500 immunity markers. And we discussed strategy for single-cell analysis using analysis of sorted cells, generated both phenotype and clonotype information, which can be useful in order to characterize full-length copies, clone them for follow-up validation studies. And we discussed that many of these assays can be run directly in the microsamples of the blood or tissue or sorted cell fraction, even without purification of RNA. And all of these technologies available as the kits or custom services. And if you're interested to use this technology, please contact us at any time, and we will be very glad to help you in your research. Thank you for your attention.